What if the next Alzheimer's breakthrough is not one dramatic cure, but a stack of smaller wins that finally start compounding? That is where the field feels headed now. We already have anti-amyloid medicines that slow decline, even if the effects are modest and the monitoring burden is real. Blood tests are moving diagnosis earlier. Delivery tools like focused ultrasound and brain shuttle designs aim to get more drug into the brain. Tau-lowering programs are trying to intervene further downstream, and genotype-led approaches are testing whether the right therapy in the right subgroup can produce bigger gains. Some of these ideas are approved. Some are in early human studies. Some have already failed. That mix is exactly why this moment matters. The field is getting more precise about what works, for whom, and at what cost. So in this video, I want to map the most credible breakthroughs in Alzheimer's medicine and the milestones most worth watching over the next few years. The first breakthrough is also the most uncomfortable one. We finally have medicines that slow Alzheimer's disease, but they come with trade-offs we can measure. Lecanemab received traditional FDA approval on July 6, 2023, and Medicare coverage is tied to participation in a qualifying registry. Donanemab followed in 2024 for early symptomatic Alzheimer's with amyloid confirmation, along with strict safety surveillance and MRI monitoring for ARIA, the brain swelling and microbleed events that make these drugs harder to deploy in the real world. In Phase 3, donanemab significantly slowed decline versus placebo. The effect is modest but real, and it is not uniform. People with lower baseline tau did better than those with high tau. That matters because it suggests the next gains may come from treating earlier and matching therapy more carefully to biology. The field is also trying to reduce the burden of treatment itself. A subcutaneous maintenance pathway for lecanemab aims to move some patients from repeated infusions toward at-home injections. So even the current antibody era is still evolving in both effectiveness and logistics. If today's drugs help only modestly, one obvious question is whether we are still under-delivering medicine to the place it needs to go. The blood-brain barrier protects the brain, but it also blocks many therapies. One strategy is focused ultrasound, which can temporarily open the barrier in precise regions. In a tiny proof of concept, combining focused ultrasound with an anti-amyloid antibody produced about 1/3 greater local plaque reduction than the antibody alone in the targeted regions. That is an intriguing signal, but it came from only 3 patients, so this is nowhere near a clinical answer yet. Another strategy is the brain shuttle idea, where antibodies use receptors like transferrin to piggyback across the barrier. The goal is simple: get more useful drug into brain tissue with the same or lower systemic dose. If that works, it could affect treatment time, practical burden, and maybe even safety. But those are still open questions. What matters today is that delivery itself is becoming a serious lever, not just a background constraint. Amyloid may start the cascade, but clinical decline tracks more closely with tau. That is why tau remains one of the most important places to look next. Tau antibodies have struggled in trials, so one alternative is to reduce tau production directly. BIIB080 is an antisense oligonucleotide designed to lower tau at the mRNA level. In Phase 1b, it reduced total tau and phosphotau in cerebrospinal fluid and showed favorable biomarker trends. The FDA granted Fast Track in 2025, and a Phase 2 study is underway. That is encouraging, but it does not settle the key question. Biomarker movement is not the same thing as preserved function. The real test is whether lowering tau early enough can translate into slower clinical decline. If it does, expect tau lowering to be layered onto anti-amyloid treatment rather than replacing it. That is the bigger picture here. Alzheimer's care may become more combined and more stage-specific, with different tools aimed at different parts of the disease process. Another major theory is that the brain's immune system could be part of the answer. Microglia can help clear damage, but they can also worsen the environment depending on state and timing. That is why inflammation drugs keep attracting attention. But recent data also show the danger of oversimplifying this story. In Phase 2, the TREM2 agonist ALO-002 engaged the biology it was supposed to engage, yet it did not slow clinical progression. That is a valuable warning. Target engagement alone is not enough. If inflammation-targeted therapies are going to matter, trials probably need tighter biomarker-defined subgroups and clearer links between a shifted signal in the lab and a real change in cognition or daily function. So I would not call this lane dead. I would call it demanding. It is the part of the field that most clearly says precision matters more than enthusiasm. One of the most practical breakthroughs in Alzheimer's medicine may not be a drug at all. It may be the blood test that gets people into care earlier. Blood-based p-tau217 assays from groups like ALZpath and Quanterix have reached analytical validation, and LabCorp launched a clinical test in 2024. In 2025, the Alzheimer's Association released the first clinical practice guideline explaining how blood-based biomarkers can fit into care. That matters because earlier triage changes everything downstream. It can reduce delays, lower dependence on PET scans or spinal taps for every patient, and make trial screening faster and cheaper. These tests do not replace clinical judgment, and they do not remove the need for confirmatory testing when results are discordant. But they could shift the whole pathway forward in time, which is exactly where disease-modifying drugs have the best chance to matter. In that sense, blood tests are not just diagnostics. They are force multipliers for the therapies already here and the ones still coming. Another reason the field may improve is simple selection. APOE4 homozygotes carry especially high risk, so some companies are trying therapies built around that biology. One experimental path is LX1001, a gene therapy meant to add protective APOE2 expression in APOE4 homozygotes. In an interim readout, the program showed APOE2 expression in cerebrospinal fluid, directional biomarker changes, and no ARIA observed in early cohorts. That is early and small, but it is enough to keep watching. On the oral small molecule side, ALZ801 is being tested in APOE4-enriched populations with the idea of targeting soluble amyloid oligomers. The Phase 3 study has completed, and the company has reported biomarker analyses supportive of its mechanism. But full peer-reviewed efficacy reporting is still pending. The common thread is not that either program is already proven. It is that genotype and subgroup selection may be one of the clearest ways to raise effect sizes without pretending one therapy will work equally well for everybody. Put all of this together, and a realistic near-future Alzheimer's pathway starts to come into focus. Earlier blood tests move patients into evaluation sooner. Confirmatory PET or CSF testing still matters when needed. Anti-amyloid treatment starts earlier, ideally with lighter burden maintenance options where available. Tau lowering could be layered in if outcomes data support it. And all of this sits inside stricter safety monitoring, registry participation, and more careful patient selection. That is not a miracle story. It's an integration story. It's also a humbling one. Not every idea works. 2 large Phase 3 semaglutide trials in symptomatic Alzheimer's failed to slow progression, which is a useful reminder that plausible biology is not enough. But the field is still moving. Watch for 3 things in the next few years. Earlier diagnosis through blood tests, less burdensome maintenance delivery, and tau-lowering results that move beyond biomarkers into functional outcomes. If those land, the everyday experience of Alzheimer's care could change in a meaningful way. Fewer chairs in infusion centers, more treatments started before the cliff, more time preserved through a series of smaller but real advances.